What if your liver's silent struggle against disease could be predicted by a genetic code hidden in its cells? Imagine a hidden battle raging within your liver, between chronic damage and sudden severe failure. That's the reality for patients with liver cirrhosis, LC, and its aggressive cousin, acute-on-chronic liver failure, ACLF. These two conditions, though distinct in symptoms, share a mysterious molecular link that scientists are only beginning to unravel. But here's where it gets controversial. Could the same biological pathways that fuel energy in healthy livers also drive their destruction in disease? Let's dive into a groundbreaking study that challenges how we understand and might one day treat these life-threatening disorders. At first glance, LC and ACLF seem worlds apart. One is a slow, creeping condition marked by scarred liver tissue. The other is a sudden, catastrophic collapse of liver function in someone already battling chronic disease. Yet, researchers noticed something strange. Both conditions seemed to whisper the same genetic language. To decode this, they turned to a powerful tool called weighted gene co-expression network analysis. Think of it as mapping out a symphony of genes, identifying which ones play in harmony and which ones clash. By analyzing vast datasets from the Gene Expression Omnibus, a public treasure trove of genetic information, the team discovered something unexpected. Genes involved in glycolysis and gluconeogenesis, key processes for energy production, were consistently overactive in both LC and ACLF patients. And this is the part most people miss. The study didn't stop at identifying these genes. It went deeper, uncovering two tiny molecular regulators, HSA-MIR122 and HSA-MIR194, that act like conductors in this genetic orchestra. These microRNAs don't just control energy metabolism, they also send signals to the immune system, potentially triggering the inflammation and immune chaos that worsen liver damage. The implications? A vicious cycle where metabolic dysfunction and immune misfiring feed into each other, accelerating disease progression. So, what does this mean for patients? The study's conclusions are bold. Targeting these microRNAs and their signaling pathways could open doors to entirely new therapies. But here's a twist that might spark debate. If these microRNAs are so crucial for liver function, could altering them risk unintended consequences? For example, HSA-MIR122 is known to stabilize liver cells under normal conditions. What happens if we tweak it in a patient already teetering on the edge of failure? The research team acknowledges this, calling for cautious yet ambitious follow-up studies. This work, published in the Journal of Translational Gastroenterology, JTG, is part of a larger mission to bridge the gap between lab discoveries and real-world treatments. JTG itself is a hub for innovation, focusing on diseases of the gut, liver, and pancreas. Its goal? To turn scientific breakthroughs into tangible solutions for patients. Yet, even as we celebrate this progress, we must ask, are we ready to embrace therapies that target the liver's own genetic machinery? Or is this a double-edged sword that demands more rigorous testing? We'd love to hear your thoughts. Do you think manipulating microRNAs is the future of liver disease treatment? Or should we stick to more traditional approaches? Share your perspective in the comments below. For the full study, visit the Journal of Translational Gastroenterology, https://www.cahepublishing.com/journal/JTG. And for a deeper dive into the research, read the full article here, https://www.cahepublishing.com/2994/JTG 2024-00047. Note, this material is provided for informational purposes and reflects the author's views. Mirage.news remains neutral on all topics, offering a platform for diverse perspectives. For the complete original text, visit Mirage.news, https://www.miragenews.com/gene signatures linked to liver cirrhosis.